The next topic that we are going to discuss under genetics is about theoretical genetics. So this one, we need to be able to use a lot of representations and examples for us to be able to teach what the concept is all about. So the first thing that we need to discuss about theoretical genetics are, of course, the definition of terms. There are several terms that are being used in theoretical genetics, such as phenotype, genotype, dominance, like dominant alleles, recessive alleles, homozygous, heterozygous, what a carrier is, what is a test cross, multiple alleles, codominant. So there are a lot of terms that is needed to be discussed. So for us to be able to convey what or explain what these terms are, we have to use examples as well. So genotype. Write down those terms first, because these are the required terms that should be memorized by students, because there are terms that are actually being required by IB. Okay, so locus, carrier, and then we also have test cross. Okay, so it's good that we are able to make a representation of each. So for example, in the case of phenotype, since phenotype, they represent the physical attributes of a specific characteristic, then it could be either like brown eyes or blue eyes. So anything that can be seen, then therefore we have to make a representation of it. And then next part, of course, in the genotype, these are the genetic constitution or the genetic representation of the phenotype. So we have to relate each and every terms together so that the student will be able to have a grasp of what they are. Okay? So representation, and of course, you have to make examples. Okay? So the next thing that is needed to be discussed in theoretical genetics is the monohybrid cross. So monohybrid cross, this is the inheritance of a single set of characteristics. So you're only crossing one set of traits in this type. So this is the most common and the most simple part wherein both SL and HL students can relate to, wherein they're just gonna use a representation of this cross using a Punnett grid. So yeah, of course, we need in the case of monohybrid cross, we need to show the genes of parents. We have to show the results or the offsprings or the number of progeny that is being produced in one cross. Progeny is also referring to offsprings. And, okay, show them using a Punnett grid. Okay. And last but not the least, we have to show the phenotypic and the genotypic ratio. Okay, like identifying the ratio of how many offsprings producing this type of trait or this type of genotype. Okay, so next, okay, we have to explain multiple alleles. So as you go along explaining what monohybrid cross is, students will find out that usually a specific gene is only controlling, a specific gene is being controlled by 2 alleles only, but it's not the case. There are also characteristics that are actually controlling 3 types of alleles, but it's just a single type of gene. So we can actually refer to that as a multiple alleles. So genes having more than 2 alleles. Okay, so students can understand further this type of this area if we give like an example in the form of ABO blood groups. So in the case of ABO blood groups, there are 3 genotypes that are there are 3 alleles that are present in the blood groups where there are the A, B, and O alleles, wherein along the way in this type of blood groups it also occurs, okay, codominance also occurs. So we have the AB blood type. So in the case of codominance, this is when a specific phenotype is actually, both phenotypes are actually affecting the characteristic. It is being expressed at the same time. Okay, so we can also, codominance can also be explained through using Punnett grid, wherein, for example, if we have a heterozygous parent having a blood type A, heterozygous blood type A, and a heterozygous blood type B parent. Okay, so if you cross them together, you would see that both both alleles are being expressed at the same time. So multiple alleles can also be explained if we are going to give examples as well, like situational examples, wherein, for example, we have like if one parent is heterozygous A and the other parent is blood type B, is it possible for them to be able to produce this progeny? Okay, so it has to be situational, because in the case of monohybrid cross, it is dependent on the product, it is dependent on the results. So students will use Punnett grid all the time. Okay, so okay, so the next part that is being discussed in theoretical genetics, we have the pedigree chart, or the family pedigree. Okay, so the pedigree chart is basically showing a specific gene or a specific characteristic that is occurring in a specific family and how it is being inherited or how it is being passed on from one generation to another. Okay, so we can also identify the pattern of inheritance of this specific characteristics, whether it is going to be showing as a dominant trait, a recessive trait, or a sex-linked trait. So, okay. A pedigree chart is an experimental cross, okay, between selected parents and the production of several progeny. So as I've said a while ago, progeny is also offspring. Okay, so a pedigree chart is showing a pattern of inheritance. So how can we show whether a pedigree chart is showing a dominant trait of warts, a dominant trait would be present if it is occurring in every generation. A recessive trait is occurring if it is going to be occurring in every other generation, or if this trait is skipping one generation and then it's going to be present the next. If it's going to be sex-linked, then it's going to affect one specific gender only. So we can show the pattern of inheritance using a pedigree chart if we use symbols, because basically a pedigree chart uses symbols only. So it uses square for males and circle for females. And then sometimes pedigree chart also shows if a specific trait is being carried by a specific individual and how this trait is being inherited or passed on to the next offspring. Okay, so identifying the dominant, recessive, or sex-linked trait. Okay, so the next part that we need to discuss under theoretical genetics is about sex chromosomes. So sex chromosomes, these are actually a pair of chromosomes that determines a gender of an individual or of an organism. So usually, okay, since they occur in pairs, they are present in X and Y chromosomes. So they control gender, and we have to explain that to students that in the case of sex chromosomes, since they are being passed on by both parents, then either parents, okay, the egg cell being carried by the mother is actually giving X chromosome as always. So an egg cell always carries an X chromosome, whereas for a sperm cell determines whether the individual is going to be male or female. So if the sperm cell carries X chromosome, then when this X chromosome sperm cell fertilizes the egg cell, then it's bound to be a female. If not, if it's going to be Y chromosome, then it's going to be male. So gender determination is always 50% chances